Hello, my name is Thomas Zambiasi. I am a senior in plant biology in the Lowry Lab, and today I'd like to talk to you a little about variation in phenotypic traits with latitude within the coastal perennial ecotype of Mimulus guttatus. So let's start by discussing some background information. Plants can have really huge ranges that cover really varied environments, and because of this, they're exposed to a lot of different climates and growing conditions and all sorts of things. So Claus and Keck and Heisey really wanted to look at this phenomenon, especially through their reciprocal transplant experiment that they did with Potentilla glandulosa. So what they did was they took plants from 3 different sites in California, from Stanford near the coast, Mather, which is mid-elevation, Timberline, which is a high elevation, and they planted them in common gardens at each of these sites. And they grew them up to assess their survival and performance at each of these sites. And what they found was that each of these plants was most suited to their home environment, including that the alpine plant was the only one to actually survive at Timberline. So they found through this that there was a genetic basis for the adaptation, but despite these genetic differences, they were the same species. They were able to create fertile hybrids when crossing these different ecotypes. So this really established that local adaptation can happen to help organisms be more suited to their home habitat. And this is accomplished through many different strategies in plants, such as flowering time, cold resistance, life history. But this brings us to our central question, which asks, does this similar variation and local adaptation occur within ecotypes that have large ranges? So now let me introduce our study system for this experiment, which is Mimulus guttatus, or the yellow monkey flower. This flower is fairly common throughout the entire western US, and is particularly common in California, Oregon, and Washington. Now this is a really great model system because there's a lot of phenotypic variation, and there are actually 2 distinct locally adapted ecotypes to investigate. One of these are the inland plants, which have annual growth, and this allows them to reproduce very quickly so they can avoid the negative effects of the yearly drought. Coastal plants don't really have this problem, so these are perennials. They survive for more than 1 year, and they use a lot more energy towards protecting the plant against things such as salt spray. Much of the research of these ecotypes focuses mostly on comparing one to the other, especially for the differences in the flowering times and the huge variety of factors, both biotic and abiotic, that can select for their locally adaptive traits. However, previous studies have actually observed that there is a variation in height among the coastal plants. So in Southern California, the plants tend to be a lot taller than those found further north, like the ones in Oregon, which are much, much shorter. And these previous studies in which this variation was observed didn't actually have sufficient data to quantify this variation, look for any sort of pattern or adaptive significance. But since the studies have been done, we've gathered a much wider range of these Mimulus seeds from the coastal ecotype, and now we have an opportunity to put together a much more complete survey of variation in height within coastal Mimulus. So this brings us to our central question for this investigation, which is, does coastal Mimulus guttatus morphology vary predictably with population latitude? So, to investigate this question, we collect seeds from 69 coastal perennial populations of Mimulus guttatus, and we planted all these seeds in small pots and weeded them so that only 1 plant would grow in each, and we grew these plants in 2 greenhouses. The first of these was at Michigan State, with 135 plants representing 56 of the original populations that were collected. And the second of these greenhouses was at Kellogg Biological Station, and there we had 402 plants representing 66 of the population. And by using these 2 different greenhouses, we would be able to show that hopefully any pattern that we could find would or would not be replicable and relatively robust to the sample size as well. And in these greenhouses, we grew up the plants, and as soon as they flowered, we measured the height of each plant, and then we calculated the average heights for each population. And so to analyze that relationship between the Mimulus height and population latitudes, we applied a linear regression model in our studio. We were also really interested in how climatic factors impacted the growth of the Mimulus plants. So to do this, we brought in wind speed data from the National Renewable Energy Laboratory Wind Prospector website, from where we gathered monthly average wind speeds at the same latitudes of each of the populations, and then from there we were able to calculate both annual and quarterly wind speeds. We also gathered data regarding precipitation, temperature, and a lot of associated factors through the WorldClim database, and brought those in for each specified population coordinates through RStudio. And because the effects of these climate variables are likely confounded with each other, just because of how climate patterns work with latitude, we applied a principal component analysis on the climate variables in each greenhouse that we performed this experiment in, and then used the first principal component, PC1, from each of these to create regressions, again using the linear regression model in RStudio. So let's get to what we actually found out from this data. So we found that in both of the greenhouses, Mimulus height did actually decrease with increasing latitude. So in the figure here at the right, the greenhouse 1 populations are represented in blue, and the greenhouse 2 populations are represented in purple. And from what we found, we can say that with approximately every degree latitude further to the north, we can predict that the Mimulus plants in those populations will be about 1 centimeter shorter on average. So we also have some plots here that show how the different climate factors correlate with PC1. So in both of the greenhouses, the factors that are the most represented within PC1 were the population latitude, the annual precipitation, the annual temperature, and also the first and fourth quarter wind speeds. So the latitude, precipitation, and wind speed were all positively correlated with PC1. So as PC1's number increases, those also increase in value, while the temperature was negatively correlated. And I'd also like to point out that the wind in general on the annual wind speed was positively correlated with PC1. It's just that the first and fourth quarter were the most strongly represented here. With these regressions here, we can see actually how PC1 correlates with the height of the plants at each population. So in both of the greenhouses, the plant height was actually negatively correlated with PC1. And this relationship was actually a little bit stronger with the larger sample size that we had in the second greenhouse. So anyways, what do these results actually mean? So we are actually able to say that there is a noticeable pattern in the coastal monkey flower height that is predictable with latitude. So as we go further north, the plants do get shorter. And because this result was actually consistent between greenhouses, all these results were consistent between both of the greenhouses, we can really be confident that this is actually what's happening because it is replicable, and also the results they hold up under the larger sample size that we had in the second greenhouse at KBS. So overall, at more northern latitudes also, where there is greater amounts of precipitation, higher winds, especially in the colder months outside of the flowering season, and with lower temperatures, the coastal Mimulus tends to be shorter. And of course, this data doesn't actually prove any direct causative factors for why the height pattern exists, and further testing is actually needed to determine what's actually going on there. So, one avenue for future study is investigating salt spray impacts. One of the things, among other factors, that really determines salt spray intensity is the wind speed. And as we saw through this research, the wind speed generally gets a lot stronger going further north. So, we also have evidence from previous work, such as that by Popovic and Lowry, that salt spray does actually harm the growth of Mimulus guttatus. So they did find that salt spray was a lot stronger on the coast, and when inland plants were transplanted to the coast, they really struggled to grow like the coastal plants did, unless they were protected by an agrofabric exclosure. So, because of this, we hypothesize that a gradient in salt spray intensity is what could possibly be driving the height climb that we see in locally adapted Mimulus. I'd also just like to give a quick thank you to Dr. Lowry for being such a phenomenal advisor as I've been undertaking this project this year. And also wanted to give a shout out to both my art discussion group in the Lowry Lab and everyone that helped out with growing plants and collecting data for this project. None of this project would have been possible without their help. And here is my literature cited, picture credits. So that is the end of my presentation. Thank you so much for listening.